Hello, I'm Dr. Aruna Saman, and in this video we are going to discuss packaging of DNA. Here is a typical mammalian cell with well-defined nucleus containing chromatin threads. These chromatin threads are called chromosomes when they undergo condensation. The approximate size of the nucleus is 10 to the power of -6 meters or 1 micrometer, whereas the length of the DNA is 2.2 meters. How then it is possible that such a long DNA molecule, 2.2 meters long DNA, gets accommodated in such a small nucleus? It is possible because the DNA molecule inside the nucleus is condensed, coiled, and supercoiled. So it fits inside such a small nucleus. But this coiling and supercoiling is different in prokaryotes and in eukaryotes. Let us first understand the packaging of DNA in prokaryotes. We will have a typical bacterial cell as a representative prokaryotic cell. The approximate size is 2 to 3 micrometers in length. They do not have well-defined nucleus. The nucleus is not well organized. The nuclear membrane, nucleolus are absent. It is nucleoid. The DNA of this prokaryotic cell in the linear condition, if we consider, then it is approximately 1100 micrometer long. But in prokaryotic cell, the DNA is never packed in a linear fashion. It is always circular and closed DNA. So, when this linear DNA becomes circular DNA, its diameter becomes 350 micrometer. Further, this circular DNA is never uniformly circular, but it is coiled and it shows many looped and domain structures. And then this coiled DNA is 30 micrometer in diameter. Further, each loop or domain is intercoiled. So we call it supercoiled DNA. And then its diameter further gets reduced to 2 micrometer. So that is the way DNA is packed in the prokaryotic cell in the supercoiled condition. Here in this diagram, we can see the coiled DNA having 40 to 50 domains or loops, and these are formed with the help of RNA connector. Whereas in this supercoiled DNA, we find that the supercoiling is assisted by histone-like anchoring proteins. There are some enzymes which maintain the supercoiled state, and these are DNA gyrase and DNA topoisomerase 1. In this diagram, we can see the RNA connector. If it is cleaved, then how the loop appears. So the loop or domain is stabilized by this RNA connector. Whereas the supercoiling of the loop is always done independently. We can prove this by the DNA getting cleaved by DNase enzyme. If the DNA domain is nicked or cleaved, then we realize that only that particular domain gets relaxed, whereas the remaining unnicked domains, they remain in the supercoiled state. So each loop is supercoiled independently. That is the way packaging of DNA is observed in the prokaryotic cell. Now let us understand the packaging of DNA in eukaryotic cell. The nucleus is very well organized. In that, the chromatin threads are seen when it is in the interphase state, and when the nucleus is in the dividing stage, the the chromosome is condensed, the length 2.2 meters approximately. And now we are going to see how such a long DNA gets accommodated in a very small size nucleus, the diameter of which is only 1 micrometer. The packaging of DNA in eukaryotes is much more complex. Here, the DNA is associated with histone and non-histone proteins. There are 5 different classes of histone proteins. These are H1, H2A, H2B, H3 and H4. These histone proteins are positively charged. It is because these histone proteins are rich in lysine and arginine residues. The 5 classes of histone proteins get organized and they form a unit of 8 molecules, which is called histone octamer. Let us see how the histone octamer gets formed by the different classes of histone proteins. Two molecules of H2A, 2 molecules of H2B, 2 molecules of H3 and 2 molecules of H4. So these 4 classes, 2 molecules each, they have contributed in the formation of octamer. Then what about this H1 protein? What is its role? Let us see. The H1 protein allows the entry and exit of DNA molecule which wraps around the octamer. So here we find that the 5 classes of histone proteins, out of which the 4 classes make octamer, and negatively charged helical DNA is wrapped around the positively charged octamer, forming a structure called as nucleosome. The DNA helix which wraps around the octamer, it takes 1 and 3/4 turns. These nucleosomes are repeated units of chromatin, and the entire structure looks like beads on string when it is observed under electron microscope. The part of the DNA between the 2 adjacent nucleosomes is called linker DNA. It has 8 to 114 base pairs. Now, this linker DNA gets coiled. As a result, the nucleosome, they come close to each other. And when 6 such nucleosomes come very close to each other, it gives rise to a structure which is called solenoid. This solenoid is seen in a chromatin. With this solenoid, the chromatin itself undergoes further coiling. And that is the way the DNA is packed in the eukaryotic cell. Let us recall the packaging of DNA once again in a eukaryotic cell with increasing complexity and compactness. In correspondence with the compactness, we find that the thickness will go on increasing. If all proteins are removed from the chromosome, then only the DNA remains, and it is 2 nanometers in thickness. We have seen that in eukaryotic cell nucleus, the DNA is never present alone. It is associated with histone proteins. So when histone proteins get associated with it, it gives rise to beads on string structure. These are the nucleosomes. And then the thickness increases to 11 nanometers. Then, due to coiling of this linker DNA, what gets produced is solenoid structure, and the thickness increases to 30 nanometers. The coiling of the chromatin to form the looped structure, that gives rise to the thickness 300 nanometers. And then so far what we have seen is the arrangement in the interphase up to the looped domain of chromatin. Once the DNA or the chromosome undergoes condensation, then supercoiling and scaffolding with non-histone protein occurs, and this arrangement further becomes compact and thickness 700. On further condensation of proteins, the metaphase chromosome has the thickness of 1400 nanometers. So this is a kind of hierarchy we find in the packaging of DNA in the eukaryotic nucleus. Now we will understand the two parts, two regions observed in a chromosome, which are euchromatin and heterochromatin. These regions are observed during interphase, when the chromosome structure is in the non-condensed state. So this is a kind of structure we observe where some region is uncondensed or non-condensed, but some region remains condensed. The non-condensed region is euchromatin and condensed region is heterochromatin. So the entire segment shows the non-condensed state, which is euchromatin, and condensed state is heterochromatin. The euchromatin stains light in color, whereas heterochromatin is staining strongly and it appears dark. The euchromatin is genetically very active and is undergoing fast replication, whereas in the heterochromatin region, the replication is almost nil. It is inactive. The transcriptionally active region is euchromatin and heterochromatin is transcriptionally inactive. You can see the amount of DNA present. It is 2 to 3 times less in euchromatin region, whereas here, due to compactness, due to condensed state, the DNA is much more in amount in the heterochromatin region. These heterochromatin regions are arranged near centromere, telomeres, and also they are intercalated. We have come to the end of our discussion on packaging of DNA. Viewers, if you have liked this video, kindly subscribe for my channel, share your comments and press the like button. Thank you.